Great. Welcome to the second place winners call for the Kaggle Novozymes competition. Jesper, I'll have you introduce your team.
Welcome, Mohammed. My name is Jesper Salomon. I'm in bioinformatics and design department in Novozyme. So it's essentially in enzyme research, and what you have been working on is also one of the molecules we've been toying around with designing. I'm actually doing mostly wild type discovery, but the rest of the team, which you actually see on the screen, are actually have been working on this. Some of this, what you have also been been playing around with. I don't know if you want to quickly tell your names, the guys in the call.
Yeah, sure.
Morten.
I'm Morten, from same department as Jesper, not working so much with protein engineering, but more in the sort of wild type enzyme space.
And Esben.
Yes, I'm Esben. I work mostly with 3D structures and protein engineering and design for process of variants for next generation products.
And finally, Peter.
Yep, I'm Peter Hallen. I'm heading a team of 6 scientists, bioinformatics and design. So we're dealing with bioinformatics, wild type enzyme discovery, and protein engineering.
So welcome, Mohammed.
Thank you.
So you can share your screen.
Yeah.
Yes, start your presentation.
Okay, so I hope the presentation is visible.
Yes.
Okay, sure. So first of all, thank you for having me. Today's agenda is a little bit about myself, and then we will be moving to the solution. So I've recently completed my bachelor's in computer science from one of the university in Karachi, and also I'm resident in Pakistan. And I'm currently working as a research engineer, and I'm very much focused on the computer vision and the machine learning part. I'm working at Retrocausal, which is basically a US-based startup, and I have around like 2 years of experience. And before joining this competition, I didn't have any protein-related experience, and it was my first, you can call it first project, which was related to protein. So like these 3.5 months was like I learned so many things and tried so many things, and some of them worked out. Moving to the solution, and looking at the overall picture, it's basically an ensemble of multiple methods, and these methods are like energy-based physical methods, and some of them are machine learning-based. So moving forward, so I've also used some of the web servers and the publicly available suite, such as these mentioned here, like SP server mod for pre-recording, and some of the software like Foldex or and all. We will be discussing them in more details in later slides. And I've also used some of the external data as well, which was shared by either by people or it was available publicly. So moving to the quick explanation of each of the method which I've used in my ensemble. The first one is the difference feature. So basically we have we were given this wild type of wild type PDB, and we were given its corresponding test mutants. So we didn't have any PDB of those mutants. So one of the user used AlphaFold 2 to generate those PDBs, and after that we can take an average take a difference of between PDB in the wild type and the mutant, and this resulted in like very good score, like that just taking a difference between a mutant and its wild type. And second, I use this Foldex 5, which is basically a software and uses this linear combination of empirical terms to calculate this energy. Then I use this Jamie. I don't know how it's pronounced, so I'm just going with Jamie. So it's basically the idea here is to use this. Basically, it uses some main parameters such as episodic. I don't know what it's called, contribution, which basically means the degree of conservation of at a particular residue. And the second parameter is this independent contribution, which tell us like how important a particular amino acid at that place is. I also use this like bunch of my ML modules with some external data, and I also provided the links here. So this data was like shared publicly at the start of the competition by this Kaggle user. And based on that data and some of the engineered features, and those features, like there were many features, and I've divided into these categories. Some of them were belonging to the structure of protein, some of them belonging to the sequence of proteins, and some amino acids, and some substitution matrix, and along with the protein BERT embedding. So protein BERT is basically a language model, and it was used to estimate the embeddings of the mutants. And we had some of the features after this feature engineering process, and these were the machine learning basic models which we were used. And the final prediction was for this machine learning method was the ensemble of all of them. Next, I use this Modfold. It's again a web server. It uses this machine learning, various techniques of machine learning, such as neural networks and like other ML techniques to predict this stability of proteins. And this PLDT is basically a B factor. So for example, when we load a PDB file using the BioPandas library, so we have this column of B factor, which is very unique to each of the residue position, and we can use it to estimate some of the energy changes in the mutant. So for example, if we are having some energy changing at position residue 1, we can, while completely ignoring the mutants, we can simply estimate with some number that at that place, at that residue position, we will be having some of the energy change here. So like if for one position, it will be like all the same numbers. So to scale them, we can use this Blossom 100 scores, which was computed using this bio library in Python. So just to scale them, and it was giving around like 0.3 score just by using the single PDB and its scores. And next is Predict Protein, is again a web server, and it basically compares the mutants or with some multiple sequence alignments, and on top of that, it uses the SVM to predict the stability. Next is RASP, which basically takes input, which is a deep neural network. It takes a 3D form of the protein. It feeds it to the 3D convolution layer, followed by a downstream fully convolution layer, and it was trained using the Rosetta values. And basically, I use the pre-trained model, which is available for free on their GitHub. Second is the RMSD, which is basically root mean square deviation. So basically, it means that basically it was computed using these two software, and this tutorial was given as well. So the basically idea here is we have this wild type protein, right? And for each of the residue, we will be checking its RMSD value. If it is less, that mean that particular residue is very stable there. So if we remove them, it will cause very instability there. So it's just an estimate that, and again, it's also a very suboptimal solution, because it's not necessarily that removing a mutant and adding a new amino acid will cause any disturbance. It's not always the case. So this is a limitation of this method, and to counter that, one of the solution is to compute this RMSD value for each of the mutants. But we were given like 22,500 the mutants, so it was not it was out of computation limitation. So that is why I could not compute that. Next is the Rosetta. I don't know how it's pronounced, so I'm going with the Rosetta. It also uses this energy-based scoring functions, some libraries, and Monte Carlo simulation to predict the stability of proteins. Next is again the Rosetta scores, but they are a little relaxed. So basically, one of the Kaggle user told in the discussion that the wild type which was provided by the competitor, by the host, was computed using the fast relax algorithm of the Rosetta. So what he did, he computed all the test mutants PDBs, and he tries to relax the Rosetta scores which we computed. So basically, it kind of improved the performance. Next, we have this SDM, is again a web server. It uses this environment-specific amino acids substitution tables, and basically the idea here is the stability is very much influenced by the structure environment. So they basically compare the mutant file with the PDB, and they use this scoring function, which has some of the you can say important features which they compare. Few of them are like listed here. Second last is the SP server is again a web server, and it also uses the scoring function. And the last was giving the very best result. So basically, again, this Thermonet is a little different from the RAS which we discussed here. It's also a C3D network, but the different component is it uses this it first of all redefine the structure using the Rosetta, and we are not only using the wild type here, we are also using the mutant here. And once we have this defined structure here, we calculate their voxel features like the ones listed here. We stack them together on top of that, and we get this 3D structure, 3D type of 3D structure of that particular protein, and same with the mutant. And then we feed it to the 3D CNN, and it just talking about the neural network, it's simply a convolutional 3D network here, followed by a dense layer, and at the end we calculate the energy. And this Servonet was also you have used like multiple external data like mentioned here. They were also like shared publicly. So our training was very straightforward. We for the web server it was simply the prediction, and for the ML models and the like the Thermonet and the other models. So some of the time was taking for training them. Some of the findings which helped me survive the you can say malicious shakeup, like you can see that there was many people who were on top and then they fall to the very like low rank. So one of the thing I noticed during the competition is that this particular range, like 500 rows to 1600 rows, I mean, based on some observations and some of the discussion on the Kaggle forum, I just estimated that they belong to the private data. So whenever I was ensembling any model, I was not only looking at its public score, but I was also looking at the distribution at that particular range. So if even if it increases the public score, but it was totally messing up this distribution, like it was adding too much noise there at that range, and then I would have discarded that method. So that is why one of the submission I chose. One was my best public model, and the second one was this 618, which placed me on the second. And on public leaderboard, it was not even my top 40 to 50 submissions. So the only reason I chose this submission because at that particular range, I had a very stable scores. Um, uh, One more thing I noticed that I mean everyone noticed that the training data was not very related to the test data, because in test data we had like single protein and its corresponding mutants. But on the training data, like we had very random data there. So, but I think that was just fine because we had like 3 months to figure out the important data and come up with the external ones and try out many things. So the method I like mentioned, these are used in my ensemble. But other than that, I've tried many other methods as well, like many web servers, many neural networks, and all. Talking about a simple model, if you see here, this 4lex was giving a fine result on public leaderboard and the private leaderboard, and same is the case with Rosetta Relaxed, and same with the Thermonet. So I think these three were the main models, the 4lex, Rosetta Relaxed scores, and the Thermonet. So I believe if we use only these three, we should be able to get around 53 score on the public leaderboard. So that was it. Please let me know if you have any questions.
Thank you, Mohammed. That was very impressive. I really can't believe you didn't know about proteins before you started this competition. That's pretty insane.
I was learning, yes, throughout the competitions.
I mean, we have some guys doing Rosetta, and they are like full-time Rosetta guys, right? So it's and you just threw it in as one of them. Did you really go through all these methods by yourself, or was it part of like the community that kind of shared the methods and then you pulled in? How did it work in practice? Including new methods.
Yes, these were also shared by the community as well. As I was participating from like very start, so I was able to discover them on my own, like the Foldex and the Rosetta. Except for the Thermonet, I tried it, but it kind of failed. But one of the Kaggle user came up with a very a bug-free solution, so I chose it from there. For the web server, I came up with my own, like by some research and all. For the rest of the methods, they were contributed by the community openly and all.
And maybe a follow-up question. You say you have these top 3 methods that are Thermonet, Foldex, and I think it was Rosetta. How much do they contribute with different predictions, or are they very much agreeing on what predictions are important?
Yes, I think they are kind of first of all, they are very diverse, right? All of them are using coming out with Foldex and Rosetta are like the energy based method, and our Thermonet is a neural network. So kind of they are very diverse. So they target different part of the predictions. And one more thing is they are very stable. So I have shown you the this public and private difference here. So you can see it's very much stable there, and not only with the high score, but there's a very little difference here. So that is that.
Okay. Yeah, there was a bit of discrepancy between the private and public, and it's funny because it's really systematically recorded, so there was no reason why it should be so different, but there was clearly a difference. It's a different region, you could say. Maybe one was closer to the active site and one was further away from the active site. Maybe that has impacted the predictions, which is interesting. Any of you other guys have some questions?
Yeah, thank you so much, Mohammed, for a great participation and presentation here also. So one question could be whether some of your work has suggested the need for additional data points for other backbones, for other enzymes. Can you comment on that? So the competition here was focused on one backbone.
Yes. Basically, the test data belong in test data we have just one protein and its point mutations, right? So we needed some relevant data, right? So we can test and train our models on them. So so that data was shared by many Kaggle users. So this is the ones listed here. Is that answered your question?
Yeah, but it was more like if there were any concrete methods that you maybe have given up on because they would require more data points or more diverse variant data coming from different backbones.
I've not tried like a lot of method based on the data, but but I believe that if we feed more relevant data to Thermonet specifically, so that should be able to like help us here more.
Okay. Thanks.
As I understood your scoring in the end, how you weighted the different methods, you looked at the private leaderboard. Can you elaborate on that? So that was could you see other people's predictions, Maggie? Is that possible? Or how did you decide on the scoring? You say you used the private leaderboard rank.
No, by private leaderboard, I mean the data range. So let me explain how I decided the weights of the assemble. So I decided them based on the public leaderboard, right? So this was just one indicator of the weights, right? So if it is performing well on the public leaderboard, then I had because they belong to the same protein, so I had this intuition that it should be belonging well to the private board. But one more thing I noticed here. In this particular range, it was also pointed out by many other competitors that we have like from 1 to 2500 rows, and these rows, even if you make some random prediction in this range, you will see this won't be affect the public leaderboard much. So so it was a hint that the private data belongs to this range. So it was also not sure, but it was just based on the observations and intuitions. So I just plotted them from 1 to 2500, and simply saw the distribution in this range, like how much the particular model which I have added in the ensemble is affecting this range here. So if it is adding too much noise, or it is like destabilizing the already made predictions, then I would not add it there.
So row is literally sites in the backbone.
No, not backbone. It was done after the training and all of that. So I mean, it's kind of a post processing, just looking
Yeah.
at the.
But you looked at the region you thought was missing, and then looked at the impact there.
Yes.
Like that. Okay, thank you.
Yeah. I also made some random predictions in this range just to know that how the leaderboard will react. But there was not any change in the score on the public leaderboard, so that is why I came up that maybe this region is belonging to the private data.
Yeah, that's a good point. Yeah. Great. Any other, maybe I have a question on the how literally did you do the ensampling? So you have a lot, you have individual contributions. Did you use, what specific strategy did you use for getting the weights of the different contributions?
Yes, basically it was kind of a trial and error method. I was thinking of this hill climbing method again and all, but I assembled these models just based on this public leaderboard and the distribution of the test data. So it was just basically an average ensemble. So when you assign some important or weights to each of the method, and at the end you simply divide them by the total weight. So for example, Forlex was performing better, I will assign more weight to it. And let's say if this SDM was not performing better, then I will assign a lesser weight to that. And at the end, let's say if I assign 5 weight to this and 2 weight to SDM, then I will be dividing them by 7, just to make them ensemble. And after the ensemble, I have also ranked them using these Spearman metrics. I don't know how it's called. Yes, it's Spearman, I guess.
Yeah, you could imagine that some of these methods are better at some regions in a protein, like, the surface of the structure versus the core. And I don't know if that could have played a part, and you kind of indirectly got that. But it's interesting. So, Esben, Morten, Marco. I think Marco also joined, which is also doing protein design in our department. Any other questions from you guys?
Just observations that you put a lot of sort of physics into this before applying the machine learning, which I, of course, as an old school protein guy, like a lot, and that might help it being more generally applicable than if you just had used something on the data alone.
Your voice was not very clear. Can you please repeat that
sorry.
question?
No, it was just an observation that you put a lot of physics into this before applying the machine learning methods, which might be why it performed so well also on the hidden data, because you sort of generalize more than if you just use the data as by itself.
Yes, true. Initially I came up with this physical methods, and it was giving fine score, and machine learning methods kind of improved their score. So yes, major portion, or you can say like 1/3 or 2/3 of them, like the score goes to this public physics method.
How much did the solution change over time? Because I noticed you were consistently in the top at least 20 over the 3 months. So it was there were a lot of others jumping in and out of top 20, but you were consistently there. So was it just because is it because you started with a physics model, or what was it that made it so high?
Yes, I think one of the reason is that I started very early, so I had plenty of time of trying like many other solutions. I mean, the ones listed here are just those who were part of my solution, but other than that, I also tried like many other web servers, methods and all. So I think that is why I was able to experiment like real good, like a lot of things there. So I think that is why.
So
I mean, I tried so many things. Yeah.
So you tried all the language models, for example. So there's no part of language models in your final example, I guess. So that didn't work so well, or what?
No, there is a language model used, the one mentioned, but it is used with the machine learning algorithm. So with the ML models, so the protein BERT embedding model. But,
okay, yeah. Yeah, ProtBERT. Yes, yeah, I know it. Yeah. We have one more question, which is a bit because we designed this. We didn't really know what to expect when we shared one data set with the community, and we have really been overwhelmed with the interest and also the activity and also you could say overfitting of this single data set. If you had to design a competition from scratch again, saying, okay, we have plenty of capacity to do more experiments, what would you suggest? Would it be more, of course, more data, but what data would be the best to make a more general model? I know it's impossible. It's not an easy question. It's an open question.
Yes. So yeah, if I had to organize that, basically it depends upon the timeline. Like let's say we are going for like 1 month or a few weeks, like 1 or 2 weeks. Then I will come up with very organized training data. Like we will be having multiple proteins and multiple protein folders, and each of them will be having some point mutations and its relevant scores. That will be very organized organized data. If you're talking about like hosting it for just 1 month and all. But if you're talking about like 3 months, I believe the training data you provided was very fine, because like coming up with solution is not always about the machine learning and all. But it's also about the cleaning the data, or researching about different methods, and actually searching for the data itself. So I think I will come up with a very similar training data, maybe a little organized and all, with some little changes, but I think that was.
So, okay, so even if you had, let's say, 10 backbones and 10 times more data, you would say you would probably come up with a similar solution, I guess, in the end. Is that what you hint at?
Yes. If I had like 3 months to host that, so yes, I will be coming up with a lot of data. So I will be expecting the competitors to like pick and point out the important ones and like ignore the rest and all, because it's a part of data science process, right? So cleaning the data and all.
Yeah.
Hi, Mohammed. Thanks for presenting and thanks for joining the competition. That was really great. I have a question now that you newly learned kind of proteins and so what are you going to do with that in the future?
Yes. As I mentioned in start, that I have recently completed my bachelor's. So in future, I will be doing the PhD. So I've not, even though I'm working in the computer vision and all of that, but I've not decided the topic yet. So maybe I can go. I mean, personally, I like this competition very much. Maybe in future, I will be going to this protein related field for PhD, or maybe a job, something like that.
Cool.
So any other questions, guys? No?
Nope.
I would just say I'm deeply impressed, and I think we all are, Mohammed, especially knowing that you didn't know anything about proteins before. It puts things in perspective. So it's very impressive how much you've gotten in 3 months' time, no less. So yeah, but congratulations. It's yeah.
Yeah, maybe just a short question, Mohammed. So do you have any overview of how much it would require to run your method, your pipeline here in a company setting? Are we able to evaluate this on our own, or how complicated would that be?
I think you should be able to do that, because so some of the web servers are used. So basically it depends upon the web server in themselves, like their availability and all. But for the softwares, I think it's more dependent upon the machine, like the one you are using. So for Foldex, I've used it on a very simple, very old laptop, so it was just fine. For one mutant, my approximate is that it should not take more than a few seconds for running this Foldex, so Seta Relaxed and the Thermonet, these three top models. So my estimate is like more than for like 100 proteins, it should take like 1 minute and a little bit more.
Okay, thanks.
There will be some licensing, Peter, for
Yes.
Rosetta and Foldex, of course.
Exactly.
Great. If there are no more questions, I just want to let you all know I included an updated presentation to the meeting request, so you can find what we just saw here. And yeah, congratulations from Kaggle as well. I'm going to go ahead and stop recording.